All right, so in this video, we're going to continue to discuss how muscles contract. And so let me share my screen. All right, and take a look. Okay, so we've been asking this question, which is how does an action potential of a motor neuron result in muscle contraction? And we talked about how there are 4 steps. And in a previous video, we went over steps 1 and 2. So now we're going to talk about steps 3 and 4. So what is happening when the muscle actually contracts? And then how does the muscle relax and return back to its resting length? So step 1 of contraction, and we mentioned previously that, if we just go back for a second, the result of excitation and excitation-contraction coupling is that you're going to have this release of calcium from the sarcoplasmic reticulum into the sarcoplasm of the muscle cell. Okay, so you have all these calcium ions floating around. And what happens, right, the calcium is going to it's going to bind to troponin, and that is going to change the shape of troponin, which changes tropomyosin, moving tropomyosin off of the active sites on actin, okay, off of those myosin binding sites on actin. So as we can see in this image, right, that has happened, right? Here are our binding sites that we can see on these actin molecules, and we can see that they are not covered. So actin is now going to be able to bind with myosin, and we're going to look at how that happens. So ATP is going to play an important role in this. And ATP is going to be bound to myosin. And we'll see where that happens. But when it's bound to myosin, ATP is immediately going to be hydrolyzed. Okay, and that means that it's going to break down into ADP and an inorganic phosphate. Okay, so ADP plus that phosphate are on the myosin head. Now, when ATP is hydrolyzed, it's going to change the shape of myosin so that it's going to activate the myosin head, meaning that it's going to go from this sort of cocked position, it's going to extend back into this extended position that you see here. Okay, so this is sort of our step 1 of contraction. ATP is going to be hydrolyzed by myosin, which is going to cause the head of myosin to extend. All right. Once that has happened, myosin is then going to bind to the active site on actin. We can see that right here. Now, this connection between actin and myosin we refer to as a cross bridge. So this step is cross bridge formation. You'll notice that ADP and that phosphate are still bound to myosin for now, okay? So that cross bridge is going to form. But as soon as that cross bridge forms, that is going to cause the release of ADP and phosphate. So, right, those are going to be released from myosin. The release of those is going to cause that myosin head to flex. So it is going to go from that extended position that this arrow is showing, right? It's going to bend forward. Myosin is going to flex. And as it does, it's going to basically sort of pull on the thin filament, right? So as it flexes in this direction, it's pulling the thin filament in the direction of these black arrows that we see here. Okay, so this is essentially causing the thin filament to slide past the thick filament. And then our step 4 is that another ATP molecule is going to bind to myosin, and this breaks the cross bridge. Okay, so that breaks that connection between actin and myosin. Okay, so we've broken our cross bridge. You can see the myosin head is still in its flexed position here. But as soon as that ATP binds, it's going to immediately be hydrolyzed again, and we go back to step 1, right? So ATP is hydrolyzed again into ADP and that phosphate, and the myosin head is going to extend. Okay, and from there it's going to be able to form another cross bridge and then flex, pulling the thin filament further, right? And so you can imagine that each myosin head, as long as there is enough calcium and enough ATP, each myosin head is just going to be continuously going through this cycle. Okay, so the cycle of contraction will continue as long as there's enough calcium and enough ATP. And remember that each thick filament, so you can see a thick filament here, here are thin filaments in our sarcomere, right? Each thick filament is made up of hundreds of myosin molecules. So it has hundreds of these myosin heads on it. And so each, as long as there's, again, enough calcium and enough ATP, you're just going to have this continuous formation of cross bridges and then breaking of cross bridges from all of these myosin heads. And at any one point, at least some of the myosin heads are going to be attached to actin. So you're always going to have some cross bridges formed at any moment, as long as the muscle is contracting. And so the end result, right, is that you're going to keep pulling these thin filaments closer and closer towards the M line, right, closer and closer towards the middle of the sarcomere. Right, so this, what we call sliding filament theory, right, is this idea that when the sarcomere contracts due to this continuous cycle of formation and breaking of cross bridges, the thin and thick filaments are not becoming shorter, right? They stay the same length, but they're sliding past each other, right? And that's why we call it sliding filament theory. All right, so that is what causes a muscle to contract. And as you have a bunch of sarcomeres contracting together, right, the entire muscle is going to contract. So then what is the process that happens when the muscle relaxes? So the first thing that's going to happen with relaxation is that the action potentials from that motor neuron that is synapsing with the muscle cell are going to stop. Okay, so the motor neurons stop sending an action potential. When that happens, it is no longer going to be releasing acetylcholine, right? So no more acetylcholine. Now, there will still be some acetylcholine left over in that synaptic cleft, in that area between the motor neuron and the end plate of the muscle cell. But the acetylcholine that is left in that gap is going to be broken down. And there's an enzyme, shown here in purple, called acetylcholinesterase that is going to break down the remaining acetylcholine. And it's going to break it down into some products, which are then going to be transported back into the motor neuron and recycled. So they'll be used to create more acetylcholine again. But as you remove that acetylcholine from the synaptic cleft, right, all of these chemically gated channels here that would bind to acetylcholine, those are going to close up when there's no more acetylcholine. And so as they close, you're no longer going to have any end plate potential and therefore no action potential in the muscle membrane. So when you no longer have an action potential in the muscle membrane, you're no longer going to be releasing calcium from the sarcoplasmic reticulum. But you still have a bunch of calcium that was released that is floating around in that sarcoplasm. And so you need to move that calcium from the sarcoplasm back into the sarcoplasmic reticulum. And that is going to be done by calcium pumps. And that requires actually active transport. So you use ATP to pump all of that calcium back into these terminal cisternae of the sarcoplasmic reticulum, where it will then stay sort of in storage, waiting for the next action potential to cause it to be, to leave the sarcoplasmic reticulum again. All right, and so as long as as that calcium gets removed, and is being sort of sequestered away again within the sarcoplasmic reticulum, now it's no longer going to be bound to troponin, and so tropomyosin is going to move back to its original position, covering all of the active sites on actin, right? Once those active sites are covered, actin and myosin cannot bind to each other, and so you no longer are able to form any cross bridges, right? And so you are no longer going to be able to produce or maintain any tension in the muscle, right? Because actin and myosin, once again, cannot interact with each other. And once they can no longer interact with each other, right, you're no longer going to be able to sort of pull actin towards, pull the thin filaments towards the midline of the sarcomere. At this point, the sarcomere is now going to return to its resting length. So it's going to sort of relax back out to how it was before it contracted. And therefore, the muscle cells are going to return to their resting length and the whole muscle is going to relax.